Hello everyone. Pulse welcomes you all to this section which is portion of science, and herein we discuss about the recent publications by the people from IGIB. We have Isha Rakheja, a PhD student from Dr. Souvik Maiti's lab today, and her recent publication, first of all I would like to congratulate her for her recent publication, which is the small molecule quercetin. It binds to MALAT1 triplex and modulates its cellular functions. So herein we are going to discuss with her about her journey throughout. Welcome Isha.
Thank you, Praveen.
So first of all, I would like to know what is the broad research area of your lab, as in what does your lab work on?
So broadly we work on RNA structure, and my work is also related to that.
Okay, so can you please tell us about your PhD project, as in what is the question that you are trying to address?
So there is a triplex motif, an intramolecular triplex motif that forms in lncRNA MALAT1 at its 3' end, and it has very important roles in the stability of MALAT1. MALAT1 is an important lncRNA because it is cancer associated. It stands for metastasis-associated lung adenocarcinoma transcript 1. So as in the name you can understand that it's metastasis associated, and the triplex imparts on MALAT1 its high stability. So my work is to try and target this triplex motif using small molecules.
All right, so like how did you reach to MALAT1? Like how did you is this something that is already going in your lab, some branch of a project that was already going on? How did you come to MALAT1?
Kind of. So Arpita, a senior in the lab, she was working on the G quadruplex motif. It is a box-like structure. It is found in DNA and RNA. So she was working on a G quadruplex motif of MALAT1, and there were reports, earlier reports, that MALAT1 also forms this triplex motif, intramolecular triplex. So it folds upon itself. So this triplex motif was not so well studied. There were only a couple of reports on that. On its structure, more reports, but targeting with small molecules, very few. And of course, the G quadruplex was also a novel report from our lab recently.
Okay. So as the title of your paper starts with small molecule quercetin, can you please let us know, like, how do we classify these molecules? Is this why do we call them small molecules, and what all molecules can be categorized into this?
Okay, so small molecules means they are low molecular weight, and many times these are derived from natural compounds. So these are natural compounds derived from like plants or similar sources, and this one quercetin is a flavonoid which we get from like berries and many other kinds of foods that we eat in daily life. And like we all often hear that this food product is good for this purpose, etc. So something is the active compound. So small molecules are low molecular weight compounds, and these, when used as drugs, they penetrate right through the cell wall, and they reach their intracellular target very easily. They can be given as oral administration of the drug, so that dose works in their case. And also you can modify them for their distribution and their mode of action. So lots can be done and has been done using small molecules.
Okay. So as I was going through your paper, I could see that this triple helix is a very small region in the MALAT structure. So is this something unique about it, or is this something that has already been known to have other functions? Like, can you elaborate on this triplex structure?
So MALAT1 is a long RNA. It's more than 8.5 kb long, and yes, you're absolutely right, the triplex is just less than 100 base pairs of it, but it's at the very 3' end, the fag end of MALAT1. So MALAT1 is processed at its 3' end, and the last portion of it is this triplex. And it forms a really interesting structure that it folds upon itself, just like somewhat you can understand, like we have shoelaces and the tips of the shoelaces so that they don't unravel. So the triplex serves a similar purpose in that it prevents MALAT1 from opening up, so the end of it from opening up. And what that does is that RNases cannot recognize or degrade it. So if they cannot find the end, they can't start degrading it.
Okay.
So as long as the triplex is formed and it's in place, then the RNA is very stable in the cell, abnormally stable, like about 12 hours or so, according to some reports.
That's a beautiful explanation. So now I would like to go on to something that everybody actually looks for, other than whatever you have already mentioned in your paper. We would like to know about your experience, as in, since most of the work for this paper might have been done during the COVID times, we would like to know what kind of problems you have faced, or what kind of technical issues, and this availability of the instruments. So can you please tell us about that?
There was some work done between April 19 to March 20, end of March, and then the lockdown happened. So the part that was done was somewhat that I could start writing up, and that helped me to think about what other experiments need to be done. So when I was able to come back to lab in the beginning of August of 20, then I did a few experiments overzealously, and then some of which worked and some did not. Then November, I was able to kind of shape it into a manuscript that we tried to submit to see kind of, what would be the response. And the journal we sent to replied back within 2 or 3 weeks to say that it was rejected. But sir then wrote to the editor, and he said that if you can answer these queries that the reviewers, so it did go into review. So the reviewers had given a lot of comments. So he said that if you can address these, then I might consider. So then I worked on those comments for a year and submitted back to them. So 21 December. So that was almost an overhauling kind of. We had worked with the shorter triplex, a 40 base pair one, which was a strict triplex, and we had removed the stem that was also part of it, because in biophysics this is quite common that you focus on the main structure and use the molecule as a probe, and it still usually answers the question. So they said that since it had been studied that the 94 nucleotide sequence was the full length, that we should work with that. So I had to repeat all the biophysics with the 94. First it was ordered, and then it came. It takes its own sweet time, like 2 months or something, for the RNA to get here, and then repeated the biophysics and all that. And then by end of 21, I submitted, and again they replied within 2 or 3 weeks that it was indeed rejected. So then, but the paper was in a much better shape according to me. So the very next day of the rejection, I submitted to the other journal, and they then took their own sweet time. They took about 4 or 5 months to reply, but this time they gave what they said was a major revision. But I already kind of knew the shape it was in and what questions they would ask. So while they were reviewing it and taking their time, I was already doing those experiments, which they finally did mention in the review. So I was almost ready with them. So they had given, I think, 3 months. So after 2 months, I submitted it back. Then after that, again, they took 4 months or something to say, but this time they accepted.
So as you mentioned about the last date in your notebook, so I would like you to give some kind of suggestions for your fellow colleagues, and the importance of how you document it, and then what all hurdles you might face when you are actually trying to compile everything, putting it up in the paper writing format, because it's very common that people say that it's just one of the hurdle gone, we have just worked for the wet lab, but it's yet that we have to sit and write. So this is another kind of fear. So some kind of suggestions you have for the colleagues.
So I don't know suggestions, but the way that I find it easy to work is that all rough work also I do in the fair notebook. There's only one notebook for me, so it has calculations on the side, and I think that comes in handy many times, because when I want to go back and refer, it makes a lot of sense that, oh yeah, I've done this calculation. So even if it was a mistake, there was the mistake. So I don't do rough where it's just one notebook with everything in it. Even if I take a day off, I write why I took a day off.
So you want to convey that it should be like a journal. So the lab notebook has to have everything that kind. We never know what we are going to use when.
Kind
So-
of like that's what I do. I think it works out because it's easy to tally that way.
Okay. So I was already going to ask you about how the publication and review thing happened. Thank you for already explaining it so well. But the next question that comes to my mind is like, what kept you going? How was it like getting on to rejection? Then even after the revision, again rejected. So how did it all go? What kind of things come to your mind? There might have been times when you would have decided, no, this is not going to work. So like how did you go through this?
No, I think the idea was novel, I would say.
Okay.
So, Sir's project, the idea behind it was definitely it had weight. So, it was not like it will not work. There were times like some experiments were stuck. A fish experiment, like visually, I was not able to see the downregulation of malat. So, but Souvik sir kept saying that if you are getting it at the qPCR level, it has to be there and you have to see it. So more in-depth analysis. I tried to do an imaging course, but it didn't really help so much. Then finally, Kaushik sir stepped in and he wrote a small script on MATLAB, and that showed exactly 50% downregulation, what I was seeing in qPCR. So it was true that it was just the right analysis that was needed. I think that was the point where I was most stuck, and yeah.
All right. So throughout, did you have some wonderful or like oh shit moments where you were like, this like I'm thankful for this, or maybe this should have not happened? Something that you would like to share?
Like not so many oh shit moments. The work stopped, and not being able to get back to work was a little bit frustrating, but I tried to make the most of it by reading up and writing, and with sir's guidance, like how to shape it into a manuscript, since it's my first publication ever. And
Big congratulations for that.
but also sir supported a lot. So wherever I would seem to get stuck, he always had a suggestion. So yeah, that's why I give a lot of credit to Souvik sir, that his guidance actually made it possible.
All right. So thank you so much, Isha. This was like really nice discussing all this with you. This is going to be definitely of great help to our audience. It was a little bit of quick, but in this greenery of IGIB, this was Isha's decision to like discuss all this here. So we really thank you and congratulate you for this paper. Thank you.
Thank you.